ended up curing bubble boy disease. But then there was an insertion of the lentivirus near a proto-oncogen which caused leukemia in a number of the children. And since that tragedy, regulators have been really preoccupied with the potential for integration of DNAs into the genome and causing insertional mutagenesis. Plasmid and viral vectors, which are some of the alternatives to messenger RNA, also can elicit innate and adaptive immune responses. And mRNA has the advantage that because we're only targeting the mRNA for delivery to the cytoplasm where it's translated, it's easier to transfect, difficult to transfect cells that do not undergo cell division. And a lot of the target cells that people are interested in to target for especially ex vivo diseases are those types of cells. And lastly, some applications benefit from transient expression. And because mRNA expression is transient, you can turn it on or turn it off by stopping to give the drug. So next I want to touch on some mRNA applications. Some of the most popular that you'll hear about at this meeting are genome engineering. So you can use messenger RNAs to express nucleases and transposons. The most popular ones recently have been CRISPR-Cas9, but there's also extensive work in clinical trials going on with zinc finger nucleases, with TALENs, and with transposons such as Sleeping Beauty and Piggyback. Most of these applications are ex vivo. In this case, the reasons to use messenger RNA, again, is that it's transient, so you're causing intentional double-stranded breaks, which would trigger integration of any DNA that's present. And so transient expression may reduce the risk of off-target effects. There's also no risk of insertional mutagenesis there because of that. You have the ability to difficult to transfect non-dividing cells, which are often the targets of these therapies. And you only require delivery of the mRNA to the cytoplasm and not to the nucleus, and so that's another advantage. Another common application is gene replacement. So this is mostly mRNAs delivered in vivo by lipid nanoparticles or polymer delivery vehicles. Again, you want to avoid the risk of DNA insertion because this is now chronic treatment. You can imagine treating these patients for the lifetime. And also some of the target cells are non-dividing cells such as hepatocytes. This type of therapy tends to be liver directed for the most part. And you can fine-tune the expression of that gene by simply changing your dosing during the course of the treatment. Another popular application for mRNA is immunobiology. And this has a number of different areas. So vaccines are very popular. So there are companies like CureVac and BioNTech and Moderna who are using mRNAs for vaccines. This is a platform for personalized medicine. So you can make a personalized mRNA vaccine for a single cancer patient, for example. These types of rapid personalized medicines, though, require rapid manufacturing turnaround times. And mRNA is ideally suited for that kind of a process. And mRNA also acts as its own adjuvant. And there are some literature that suggests that you get activation of different MHC pathways when you express an mRNA intracellularly versus expressing or delivering a protein extracellularly. And so it may have some advantages. Another interesting area here is the area of self-replicating RNA or replicons. These are often based on viruses such as alpha viruses. And you can give a very low dose because the RNA, once it gets inside the cell, goes through replication cycle, although it doesn't produce active virus particles. And by going through a double-stranded intermediate, that activates immune pathways and it acts as an adjuvant. Again, people are interested in these kinds of things for responding to pandemics or bioterror incidents. And so rapid manufacturing times are also critical here. And a last one that I want to touch on is the inactivation of checkpoint inhibitors using CRISPR. It's another popular application of messenger RNA. All right, so now I'd like to spend a little bit of time telling you about how your body recognizes self-RNAs and distinguishes them from non-self-RNAs. So as Jim alluded, there are several characteristics that distinguish mRNAs that are delivered externally from those that are internal. So mRNAs are found in the cytoplasm or the nucleus. They're generally not supposed to be found in the endosome. And so the cell recognizes mRNAs in that compartment as being potentially viruses. There's also several characteristics that are added to the RNA during the processing events, such as addition of a cap and methylated caps that also distinguish it and license it as a self-RNA. And also, self-RNAs tend to lack long double-stranded structures that are found in viruses. And there are certain viral structure elements which are detected by the innate immune system. All right, so the endosomal innate immune receptors, or we call them pattern recognition receptors, are primarily toll-like receptors 3, 7, and 8, which recognize single-stranded and double-stranded RNA. And these are basically virus sensors. Now, depending on your platform that you use for delivery, you may transit through different compartments, and this is important. So when you're looking at people's data about certain biology, keep in mind if you're using a different delivery vehicle. So for example, you're using an endosome, you may be a liposome, you may be trafficking through an endosomal compartment, whereas you're doing an ex vivo electroporation of some CD34 positive cells, you may bypass that compartment. And so the delivery vehicle that you use is going to depend, is going to impact what characteristics your messenger RNA might have. All right, so there are also a number of cytosolic receptors that recognize mRNAs in the cytosol. And these would include PKR, which recognizes short double-stranded RNAs, MDA5, which recognizes long double-stranded RNAs, RIG-I recognizes 5' triphosphates. There's a family of proteins called IFITs, which recognize 5' triphosphates and cap0. And then lastly, there's a newly discovered pathway called cGAS-STING, which recognizes cytosolic DNA ends. All right, so now I'm going to shift gear and talk about the manufacturing design considerations for a messenger RNA. So what would the optimal mRNA look like? Well, we want the activity and duration of expression to be tailored to the application, whether that's chronic or transient. We want it to be possible to produce the RNA with financially attainable costs. The template should be designed for ease of manufacturing. And the immunogenicity or the lack of immunogenicity should be dictated by the application, whether that's a vaccine or a genome editing event or whatever. So synthesis and purification processes should be consistent and enable consistent high-quality compounds. And you want to take into consideration the end game as you're designing your process. Plan ahead early for the scale-up of your mRNA, because you may find that the manufacturing processes that can be used at small scales may not be scalable to the scales you might require for your particular application. So think about those kinds of things well in advance. So this is how, in general, we synthesize messenger RNAs. So we use a bacteriophage polymerase called T7 RNA polymerase. And that T7 polymerase reads a transcription template, and that transcription template dictates the sequence of the mRNA that's going to be produced. In that reaction, you can have standard or modified NTPs, and optionally, you can have some cap analogs to do co-transcriptional capping, and I'll talk about that in a later section. A couple optional pieces are inorganic pyrophosphatase and RNase inhibitor. So during the manufacturing process, the first thing we do is we think about the template design and construction, and then we linearize the template. You do in vitro transcription with the T7 polymerase, and then you may include a DNA step to degrade the DNA template. There's a number of post-transcriptional modifications which are possible. For example, if you have not co-transcriptionally added your cap, you can do that enzymatically. You can 2'-methylate the RNA to form what's called a cap1 structure, and we'll talk a bunch more later on about cap structures. And you can add a poly-A tail if it wasn't templated. In general, we're doing a purification of the RNA, and then you can have some optional treatments, for example, a phosphatase treatment to remove residual triphosphates. There's generally in-process assays, and then sterile filtration and final release and fill. So during the research phase, the client is generally providing the selection of the target. At that point, the open reading frame is designed, and we'll talk a little bit more about that later. We'll select the 5' untranslated region upstream of the start codon, 3' untranslated region downstream of the stop codon, and this is the open reading frame or the coding sequence here. So at this point, we'll select whether or not we want to use wild type or modified bases, and this may require some experimentation in your model system. We'll determine the polyadenylation strategy and the appropriate purification method. And as Jim alluded to, these mRNAs are structured with caps and poly-A tails. And the function of these caps and poly-A tails and untranslated regions are to bring together the 5' and the 3' end of the RNA and make a little factory where the ribosome's going around and around and around in a circle making proteins. So this is basically what a minimal plasmid looks like. So I just wanted to go over some of the elements of the plasmid. So in general, these are bacterial plasmids with an origin of replication. They'll sometimes have an antibiotic resistance gene because people are allergic to ampicillin, like myself. People tend to stay away from that selected marker. It has a 5' untranslated region, the coding region, and it has a 3' untranslated region, and then usually a small poly-A tail. And then you'll also want to have a linearization site because the transcription functions by runoff transcription off the end of the message. Now, in terms of adding your poly-A tail, you have a couple different options. So you can encode the poly-A tail within the plasmid. The difficulty with that is that long poly-A tails are not stable in bacteria in general, and so you get recombination events. It's fairly easy to get an 80-base poly-A tail to be stable in bacteria, but shorter tails may have lower activity, and then maybe something open for discussion after Jim's talk. This may be acceptable in some applications, but it's the most affordable approach. Alternatively, you can use an enzyme called poly-A polymerase to add the poly-A tail. This can give you much longer poly-A tails. We've seen poly-A tails in the thousands if you really want to push it. But there's a significant cost component to poly-A polymerase, and it gives a distribution of poly-A tail lengths. And so you have to worry then in a manufacturing process about the reproducibility of the poly-A tail length addition. And so in many cases, we've had clients ask us to develop a quantitative or qualitative poly-A tail length assay. And here's just an example of what one kind of looks like. So this is just a bioanalyzer trace of an RNA before the polyadenylation event. Again, it's a distribution of sizes with the maximum. And then after polyadenylation, you see you get a distribution of different poly-A tail lengths, and we can take the peak of that peak and compare it to the peak of the original peak and get an idea of the difference in the distributions of those two populations. There are also some other more accurate methods that we can develop for you too as well if you need something more quantitative than this. So in the next section, I'd like to talk about mRNA capping. So just to give you a little bit of an overview of the eukaryotic mRNA cap, it has an inverted M7G here and a triphosphate linkage. And then the thing I'd like to focus on is these 2' positions of these sugars at the first base and the second base. So when there's no methyl group here, it's just an OH, we call that structure cap 0. Cap 0 is an intermediate in the production of a cap, but it's not found at all in any eukaryotic mature messages. So 100% of eukaryotic messages have a methyl group at this first position, and we call that cap 1. And about 50% of mRNAs have a methyl group at this point, it's called cap 2. And so we think that nature thinks that these two methylation positions are really important. There's also another additional cap that's been recently appreciated, which we call M6AM. And so that is at the N6 position of adenosine. Here, there's an additional methylation event in addition to this methylation to make cap 1. And there's a really nice paper that I recommend to you from our collaborator, Sami Jaffrey, in Nature recently, where they showed that M6AM was very prevalent, up to 30% of caps in eukaryotic messages, and that they had evidence that it regulated translational initiation and mRNA stability. And I'll tell you a little bit about that and show you some data a little bit further on in the talk. So what are the functions of these caps? So caps are known to be functioned in nuclear export. in splicing, turnover, protein synthesis, decapping. They're also thought to be important for self, non-self recognition. So Cap0 is recognized as foreign, and there are proteins, for example, IFITs, which recognize improperly methylated caps. That binding of these IFITs reduces initiation by translation initiation factors. Cap1 methylation also modulates the binding to these pattern recognition receptors that I told you about earlier in the talk. And the role of Cap2 is largely unexplored because it was not possible to easily generate Cap2 RNAs until very recently, and I'll show you some methodology that we developed at TriLink to do that. So basically, we don't really know what Cap2 does, which is exciting. All right, so viruses think that the cap is important. So many viruses possess a Cap1 structure, especially cytoplasmic RNA viruses. And many viruses that replicate in the cytoplasm also encode a methyltransferase, which is adding that methyl group to make the mRNA Cap1. There's for certain viruses that actually go and steal the caps off of eukaryotic messages and splice them onto their own messages to achieve Cap1 structures. And when you block the methyltransferase activity of many viruses that are RNA viruses in the cytoplasm, that blocking of that formation of Cap1 leads to attenuation of that virus. So all of this suggests that the capping state of the mRNA is important and that viruses have gotten smart to this. So there's a series of proteins called IFITs 1 through 5, and these recognize RNAs with a 5' triphosphate or Cap0, and that they bind to them and sequester them from the translation machinery and compete for binding by cap binding protein. Now, interesting, it's been shown that IFITs reduce binding, or have reduced binding for Cap1 and Cap2. And for example, when you take a pox virus or coronavirus, the host restriction to that virus is blocked by Cap1 methylation. So if you methylate those viruses, the cell becomes permissive for those viruses. The most recently studied group out of the IFIT group is IFIT5, and it was shown to bind to 5' monophosphate, 5' triphosphate, and Cap0, but not to Cap1. So the capping state of the mRNA is determining which of the pattern recognition receptors in the cell that are binding to these different RNAs. So there's another pair of cytosolic sensors called RIG-I and MDA5, and they also care about Cap1. So RIG-I is not activated by Cap1 double-stranded RNAs, and the attenuation of coronavirus replication by blocking of methyltransferases is relieved in the absence of MDA5, suggesting that it plays a role in MDA5 binding. All right, so in the next section, I want to tell you about methods by which we can cap RNAs. There are basically two classes of capping methods that we use for capping messenger RNAs. The first category is called enzymatic capping, in which we make the RNA and generally purify it, and then use vaccinia virus enzymes to cap and methylate the messenger RNA. The second category is co-transcriptional capping. And there are 3 generations of cap analogs that I just wanted to point out to you. The first generation was cap or MCAP, and that worked kind of okay. Then there came kind of a revolution in this area, which was something called anti-reverse cap analog, which I'll tell you about a little bit later. And this one worked much better than the first generations, and this has kind of been the industry standard for a couple decades, really. And most recently, we at TriLink developed a new method which we call clean cap, which we think is going to be the 3rd generation of capping, and I'm going to tell you about that in the next few slides here. But first, I want to tell you a little bit about the characteristics of the other capping methods. So enzymatic capping utilizes a vaccinia virus capping enzyme and a 2' O-methyltransferase. And you can get 100% capping if you drive this reaction to completion, in most cases. The yields are a natural, unmodified cap structure, which may be good, And the Cap1 structure is less immunogenic in vivo. And so this gets you to a desirable capping state. But capping enzymes are costly at scale, and there's significant batch-to-batch enzymatic variations. They also use an unstable cofactor, which is S-adenosylmethionine. And the capping efficiency can vary, especially if you have a structured 5' end, which is often the case with these replicon RNAs I told you about earlier. You can get inefficient capping if you have a very strong structure at the 5' end, or you get variability. So in order to assess capping, we developed a capping assay that I'll walk you through in just a moment. So as Jim mentioned at the beginning, we're trying to assess the capping status of an RNA that may be millions of Daltons in size. And we're trying to distinguish a single methyl group in that sea of millions of Daltons. That's really difficult. So in order to do that, we utilize a strategy where we made a DNA zyme and cut off the very end of the RNA at a very specific location. And then we analyze that by LCMS. Sometimes we'll include a phosphatase step to simplify the analysis of mono, di, and triphosphates. And over here on the right-hand side, you can see an example of an enzymatic capping reaction that's gone most of the way, but not entirely to completion. We're able to see an uncapped monophosphate, cap 0, and a little bit of triphosphate. And so this is what it looks like when an enzymatic capping does not go to completion. It can be much worse. This is actually a pretty good capping. All right, so the second possibility is to use a co-transcriptional capping strategy. And so I told you that the industry standard for the last 20 years or so has been ARCA, and it's called anti-reverse cap analog because it has a 2' or 3' O-methyl group here. And what that does is that prevents this RNA from elongating from this side. So this construct cannot go in backwards. It's kind of almost a symmetrical molecule. This forces it to go in the right direction, and that was the innovation of ARCA. Now, during a normal transcription initiation event, it looks like this. So you have a T7 promoter, and here at the edge of the blue circle is where the transcription starts, at the +1, +2, and +3 nucleotides outside of the promoter. Normally, you would initiate with GTP, but instead, you can initiate with this cap analog here, where again, you're initiating with G at the +1 position, but you have this all hanging off the end, and the polymerase seems to be okay with that. Right? But these two reactions compete with each other, and so generally what you do is you use a large excess of the cap over GTP, such that you get capping, because when you initiate with this, you get a triphosphate instead of a cap. And so in general, you're getting somewhere around 70 or 80% capping when you're using, say, a 4 or 5-fold excess of this. But this is the most expensive reagent in the capping reaction. So what are the characteristics of ARCA? Well, it yields cap 0, and I told you earlier that we think that might not be such a good thing. You have a competition between GTP and the cap analog, and so you have to use large excesses of the cap analog. It gives you fairly reproducible capping, so that's good. So in general, you can get about 80% capping. However, there's significant amounts of transcriptional stuttering, which I'll show you in just a second. And the cap contains an unnatural 3' O-methyl group, which may be good or may be bad, something you would have to test. Now, if you get 80% capping, the rest of that material is 5' triphosphate, which we told you has the potential to be immunogenic. And so sometimes we'll include a phosphatase step to remove this residual triphosphate. And then in general, transcription by T7 polymerase likes to start with a G. It highly prefers a G. And so you're constrained in terms of the sequence you can use at the 5' end of your transcript. So this is just an example of a capping assay, a very typical one for ARCA, where here you can see the uncapped material is substantial. And then this is the cap 0 material, and you get a lot of transcriptional stuttering. So this is the addition of untemplated nucleotides at the beginning of the transcription reaction. And we see this is a very typical kind of ARCA capping reaction. So in order to get around some of these obstacles, so the stuttering at the beginning, the inability to make a cap1 structure co-transcriptionally, we came up with the idea of, instead of using a dimer to cap, we'd use a trimer. And so Here you can see the M7G and then 2 bases. These sit down at the +1 and the +2 position. And this allows us to bring in a methyl group here to make cap1 co-transcriptionally. And this actually caps extremely efficiently. We can also make a cap2 structure by using a tetramer, where we bind to the +1 and the +2 and +3 position, bringing in 2 methyl groups. And this allows us to achieve cap2 structures. And again, this is very early days. We're just getting to the point of learning how to make these things, and we basically don't know yet what they do. So what are the cap2 structures? And again, this is very early days. We're just getting to the point of learning how to make these things, and we basically don't know yet what they do. So what are the characteristics of clean cap? So you get efficient capping that's highly reproducible. With our best clean cap analogs, we regularly get in the very high 90s. For some of the more challenging ones, more like 90, depending on the sequence at the beginning. We can produce cap 0, cap 1, or cap 2. We can yield unmodified natural caps, or if you want one with a modification, we can put a whole bunch of different chemistry there. So it opens up the chemistry at the 5' end of your RNA. Modified natural caps, or if you want one with a modification, we can put a whole bunch of different chemistry there. So it opens up the chemistry at the 5' end of your RNA. And it also allows us to put things like M6A or M6AM at the beginning. And I'll show you some data later on in the talk about that. It's much more cost effective than enzymatic capping. It's easily scalable. And one of the things we like about it is that in an ideal manufacturing world, you would have a synthesis where you do a one-pot reaction, you go to a purification, you filter, you're done. And so being able to co-transcriptionally cap and achieve cap 1 and cap 2 structures opens the possibility of a one-pot reaction, as long as all your downstream stuff is compatible with that. So this is an example of a clean capping. This one went really well, and the capping is in probably the 99% range. Here's one that went slightly less well. But we get extremely reproducible and very high levels of capping with our kind of first generation clean caps. And this is just a montage of the 3 different capping methods, so you can kind of see them sort of side by side. We have ARCA with a lot of uncapped material and a lot of transcriptional stuttering. We have enzymatic capping, which has some incomplete capping here. And then we have clean cap, which this is the incomplete capping in the clean cap. And so we're very, very excited about this new technology. And so we've been applying it quite heavily, and we've gotten really nice adoption from the community with it. All right, so now I'm going to change gears and talk a little bit about chemical modification of messenger RNA. So chemical modification of RNA was a topic that basically was kind of put forth by Katie Carrico and Drew Weissman quite a number of years now in a series of really nice papers that I'd recommend to you. And what they showed was that modification of mRNA with different modified bases, especially with pseudouridine, reduced bindings to innate immune sensors in vitro. It reduced toxicity and prolonged the expression in cells, and it increased translation in vitro. And so for quite a while, pseudouridine has kind of been the most popular modification in the arsenal. So because TriLink offers over 200 modified NTPs, we wanted to ask, well, can we find other modified bases that have desirable biological properties? So we embarked on a project where we synthesized numerous naturally occurring and unnatural modified NTPs. And we asked, can we identify an analog that's superior or equivalent or superior to pseudouridine? And so we put these modified bases into a couple different templates. So originally we started off with EGFP and luciferase. And the most promising modifications were tested in additional sequence contexts. So the first thing we did was to measure whether or not they could be incorporated with T7 polymerase, because if the polymerase won't take them, then it's not going to be very useful. And so that eliminated a bunch of NTPs. Then we measured their translational capacity in an in vitro translation system, and then in 6 different cultured cell lines. And later we examined the effect of sequence engineering on transcription yield, quality, and the immunogenicity of these transfected mRNAs. This is just a smattering of some of the different modified bases that we tried. We tried a bunch more in addition to this. And so here's the case study of what the results were with these messenger RNAs. So first, the in vitro translation. So this is a translation in a rabbit reticulocyte lysate. And you can see that wild type, so A, C, G, and U, wasn't the one that was translated the most efficiently in these lysates. In fact, pseudouridine was translated at a much higher rate in the rabbit reticulysate than wild type RNA, which is sort of surprising because this is the substrate of the ribosome, the endogenous substrate. So there must be something more going on there. And we think actually has to do with immunity. We tested these in a number of different cell types. This was a really nice collaboration with Joel Jesse at MTI Global STEM, which is now part of Thermo. And they used their Global STEM mRNA-in reagent, which is a cationic lipid nanoparticle. And so when you're thinking about the results of this, you might think, okay, well, I told you earlier that delivery is important. You might not compare these results directly to, say, an electroporation. Okay, so this is with the lipid nanoparticle. And this is some of the data, and there's a lot of data here, and I don't need you to go into detail. I want to make some really simple points here. One point is that if you go along the different cell types, the pattern of which modifications are tolerated is different. So this cell is much less permissive than, say, this cell type. Okay, so permissiveness to different nucleic acid modifications is dependent on the cell type. And this first example here I'm telling you about is in the GFP sequence context. So we made the whole series that we'd made in GFP, and we now put it into luciferase. If you just take a look at this picture and contrast it with the next one, what we found was that the pattern was different. Okay, so there's a sequence context effect and now a cell type context effect. So we started asking ourselves, well, what's different between these two primary sequence contexts? And we noticed that EGFP had very few number of contiguous uridine residues. And so we wondered whether or not that might be part of the answer. And so we were in the middle of doing this study where we're kind of screening through a number of different pseudo-U homologs, and we made some interesting observations. So these get bigger as you kind of go down the row here, and we found that if you look at just the first bars, the dark bars, that as we went to larger sizes, the ability to incorporate into the transcript was diminished. However, but in EGFP, which has fewer uridines, we saw that we could incorporate some of these larger modifications into GFP that were not incorporated into luciferase. And so then we asked the question, if we depleted the uridines in the luciferase sequence by using substitution of synonymous codons, would that improve the incorporation? And you can see if you now look at the blue, lighter blue bars, that we are indeed, so this is with lots of uridines, and this is with fewer uridines again here, that we could change the incorporation based on depleting the sequence of uridines. And this is something that other people in the field have looked at. For example, CureVac has published some papers using something that they call GC enrichment, which is not exactly the same thing, but gives you basically the same result. You're enriching for G's and C's, you're depleting for U's, right? And they see some interesting things along the way. I suggest you read their papers. All right, so here's just a bioanalyzer trace where you can see that this mom substitution was not easily incorporated at all, but now when we do the uridine depletion, we can now incorporate this larger modification. Now, when we looked in wheat germ extracts, we saw that uridine depletion here on the right-hand side versus the left-hand side gave us more activity. So where we were seeing no activity here, we were seeing some activity here. But when we looked in cell culture, we saw really dramatic effects. So here on the left is non-uridine depleted, and here on the right is uridine depleted, and we had to cut the scale in order to be able to put them both on the same graph. And so we saw significant improvements in activity upon uridine depletion of the sequences. And you can see here that, a lot of these sequences, so here's pseudo-U now. And we actually have things that are working better than pseudo-U. We know N1-methyl pseudo-U has also been reported in the literature, but we have other things like 5-methoxy-U, which is also working pretty well in a uridine-depleted context. All right, so next I want to give you a little case study that we did with Matt Porteous's lab at Stanford. So Matt Porteous is one of the first people who did genome engineering experiments way back with David Baltimore using zinc finger nucleases. And so we did a nice collaboration with him where we looked at Cas9 and put different modified bases into a standard Cas9 or a uridine-depleted Cas9 sequence. So the standard Cas9 sequence we'd gotten from our collaborator Feng Zhang, and it's a sequence that many of you might be using. And then the uridine-depleted sequence, we removed as many uridines as we could. These were electroporated into CD34 positive primary human T cells, or primary cells. And then we measured indel formation using what's called a tide analysis. And again, this is in collaboration with Matt. And so here you can see a series of different modifications, and we're comparing it to a ribonuclear particle delivering a guide strand. And again, you can see that when we uridine deplete the sequences, we get higher activity. Interestingly, in this sequence context, we see that wild-type RNA, which doesn't always behave well in other contexts, was working as well as 5-methoxy-U and pseudo-uridine in this context. Now, this may be because we're electroporating the construct. I'm not sure. But this was what the observation was here. You can see this is wild-type when it's not uridine depleted. And the pseudo-U was pretty good either way. So uridine depletion seemed to help the wild type, and also the 5-methoxy-U were improved by uridine depletion. So some conclusions about the chemical modification section is that in vitro translation does not correlate with cell activity. And so that suggests that there's another layer on top of that, and we think that that has to do with innate immune activation. Primary sequence selection is important for optimal chemical modification. And we found in a number of different contexts that uridine depletion frequently improves incorporation by T7 polymerase, as well as their yield and quality. And that in numerous sequence contexts, we found that uridine depletion in the combination with 5-methoxy-uridine yields high cell activity, low toxicity, and reduced interferon. In the context of CD34 positive cells that have been electroporated, uridine-depleted Cas9 mRNAs, the wild-type pseudo-uridine and 5-methoxy-U RNAs all yielded similar indel rates. And I just want to make the caveat that I'm not saying that that would necessarily be true if you were doing a lipid transfection with the exact same constructs. We didn't test that. So I think delivery context also matters. Okay. So another case study here is on the optimization of mRNA purification. So why are we purifying the messenger RNA? We're trying to get rid of a lot of the ingredients in the process, like salts, NTPs, cap analogs, the proteins that were used in the reaction, truncated mRNA products, residual DNA, and we think that the removal of double-stranded RNA can be really important. Traditional approaches such as silica membrane purification or precipitation are what people have generally used. But scalable purification may be the hardest part of this whole puzzle, how to do that. Making the mRNA is actually not all that hard. So we've done up to gram scale syntheses and purifications using silica membrane techniques, but I don't think we'd really want to go past about a gram. And so we thought, well, most commercial biomolecule production relies on process chromatography. And so we evaluated a large number of different chromatography approaches. We have one that we call LC isolation, which is good at removing residual proteins, DNA, and NTPs, but not really double-stranded RNA. We have another process, which is a reverse phase HPLC, that can be pretty good at removing double-stranded RNA, and it enriches for full-length RNA. It becomes